Hello friends, so in this video we're going to be studying the nitrogenous bases as per our studies on nucleic acids. In the previous video we studied the pentose sugar and the phosphate group as components of our nucleotide which forms our nucleic acids. Now in this video we're going to be studying the nitrogenous bases. The nitrogenous bases are molecules that possess nitrogen in different amounts and they function as a base. I repeat, the nitrogenous bases are molecules that possess nitrogen in different amounts and they function as a base. In the nitrogenous bases, nitrogen combines with carbon, oxygen, and hydrogen in varying proportions to form ring-like structures. The nitrogenous bases are called bases now because they possess the ability to donate electrons to other molecules and also form new molecules in doing this. They possess the ability to donate electrons to other molecules and also form new molecules in doing this. Now, we say that the nitrogenous bases are ring-like structure, right? They are ring-like structures. So there are two basic types of rings that the nitrogenous basis forms. And these rings separate the nitrogenous bases into two types. Now, the first type of nitrogenous bases are called the purines. The first type of the nitrogenous bases are called purines. And the second type are called pyrimidines. The second type are called pyrimidines. The purines are double-ringed structures. The purines are double-ringed structures, while the pyrimidines are single-ringed structures. The purines are double-ringed structures, while the pyrimidines are single-ringed structures. This right here would be a good example of the double rings of the purines. And the pyrimidines are single-ringed structures. This right here would be a good example of the structure of the pyrimidine. Now, there are two types of purines now, and these are one, adenine, and the second is guanine. There are three types of pyrimidines. The first one is cytosine, the second is thymine, and the third is uracil. These are the types of purines and pyrimidines we have. Here are some of the structures of these purines and pyrimidines. Here are the structures of the purines and the pyrimidines that we listed above. Now, the purines we have in blue and the pyrimidines we have in green. Now, notice now that the purines, adenine and guanine, they are double-ringed structures. They are double-ringed structures. Also, the pyrimidines now, they are single-ringed structures. You have the cytosine, thymine, and the uracil. Due to the double rings of the purines are larger structures than the pyrimidines. The purines are larger structures than the pyrimidines, which are just single rings. These are the nitrogenous bases that would help us understand the concept of our DNA and RNA structures, which we said are nucleic acids. Now, recall that we said that the nucleic acids are formed from repeating units of nucleotides, right? So, in understanding that, we had to study each of the components that form the nucleotides. And we said that the nucleotides are formed from the pentose sugar, the phosphate group, and the nitrogenous base. And each of these components we have studied in details. Now let's see how each of these components combine together to form the nucleotide. Let's study how each of them are bound to one another to form the nucleotide. Here is a classic example of the structure of a nucleotide. You have the phosphate group, you have the pentose sugar, we have the nitrogenous base. How true, how true. Recall now that in the previous video we said that in numbering the carbon atoms of our pentose sugar, we number in a clockwise direction. So we have 1, 2, 3, 4, and our fifth carbon atom will be outside the ring, and there you have it. That's our fifth carbon atom. Now, in binding, the phosphate group is bound chemically to our fifth carbon atom. It's bound to our fifth carbon atom. And this bond is known as an ester bond. It's known as an ester bond. How true, how true. Also, our nitrogenous base, in binding now, it's chemically bound to our first carbon atom. Is bound to our first carbon atom. And if this nitrogenous base is a pyrimidine, it's going to make use of the N1 nitrogen atom in binding to our pentose sugar. I repeat, if this nitrogenous base is a pyrimidine, which are single ringed, right, it would have to use the N1 nitrogen atom in binding to the pentose sugar. But if this nitrogenous base is a purine, let's put a purine right there. That's a purine right there. Now, this purine now is going to bind with our pentose sugar using the N9 nitrogen atom. It's going to bind with the pentose sugar using the N9 nitrogen atom. And the bond between the purine or the pyrimidine now, our nitrogenous basis, and the pentose sugar is called a glycosidic bond. The bond between the pentose sugar and the nitrogenous basis is called a glycosidic bond. This is how the three components of a nucleotide bind together to form the nucleotide. Now let's study how many nucleotides bind to one another to form a chain of nucleotides called the polynucleotide, which forms our nucleic acids. Now, if we study this structure from below, we'll notice like that, just like we said before, the phosphate group attaches to the carbon number 5 of this nucleotide below. This same phosphate group then attaches to the carbon number 3 of the nucleotide above in this manner, and this repeats itself all throughout the length of the polynucleotide. So there is the attachment between the phosphate group and the carbon number 3. This forms a long chain of polynucleotide, which is made up of a sugar phosphate backbone. A sugar phosphate backbone right here, down through the length of the polynucleotide, which forms our nucleic acid. Now, this structure right here, if the sugar forming this structure was a ribose sugar, this would be the structure of our RNA. Assuming this sugar now forming this structure was a ribose sugar. How is this so? Because the RNA now are single stranded structure. But in order to form the structures of the DNA now, what happens is that a similar strand of polynucleotide runs in an opposite direction to this strand, just like we have in this diagram. So the second strand runs in an opposite direction to the first strand. Notice now that the nitrogenous bases are pointing towards each other at the center, and they are bound chemically by hydrogen bond. A hydrogen bond here, a hydrogen bond here also, and another hydrogen bond here again, another hydrogen bond here again. But take note, in the bonding of the nitrogenous bases to one another, they have to obey the rule of complementary base pairing. They must obey the rule of complementary base pairing, whereby adenine always binds to thymine. Adenine binds to thymine, and cytosine always bind to guanine. This is repeated all throughout the structure. So we have thymine, adenine, we can also have guanine and cytosine. So whichever order it takes now, the adenine always binds to thymine and cytosine always binds to guanine. However, scientists envisage that the DNA molecule is somewhat of a twisted ladder, whereby the two uprights of the ladder, as you can see in the diagram, represents the sugar phosphate backbone, and the rungs of the ladder represent the nitrogenous bases sticking inwards towards each other and are linked up in specific relationship, adenine binding with thymine and cytosine binding with guanine, and this process repeats throughout the length of the structure. And of course, the DNA molecule now is actually a double helix structure. This was proposed by Watson and Crick in 1953. Here is the double helix structure. This is the double helix structure, whereby the helices represent your sugar phosphate backbone, and at the center you have your nitrogenous base in pairs. That's my time, guys. Thank you so much for watching. See you in the next video.